So, hopefully you're still with me. We have gone through the physiology of the nervous system and how we sense pain. We talked about how that pain gets transmitted up to the brain. And we've talked about kind of the theories that help us explain how pain is transmitted and felt. Now we're going to talk about how we actually control pain and the levels of pain control. So we're going to have 3 levels that are based on the various theories that have come out. Okay, specifically dealing around gait theory, the ascending and descending mechanisms, as well as a little bit on the central control theory as well. So, first one we're going to talk about is what we call level 1 pain control, or it's commonly called gait control. It's mainly the ascending mechanism of gait, of the gait theory. So the idea is to close the gait, okay? It's this ascending pathway, and we talked about this already, that we have this stimulation of non-painful nerve fibers can actually potentially block the transmission of noxious stimuli, of painful stimuli, okay? So the pain inhibition takes place in the periphery. So again, it's an ascending mechanism. It's coming from the periphery and happens at that substantia gelatinosa, the SG, inside the dorsal horn of the spinal cord around lamina 2. Okay, and the A beta fibers get stimulated, and that prevents the A delta fibers and the C fibers from being able to transmit their signal past that T cell. Okay. Kind of that gating mechanism, and they're not allowed to get through because the signal is being overwhelmed with the A beta signal. It's what we call a presynaptic inhibition of the second order neurons. So think about what that means. Before the synapsis, so before the connection between our first order, the afferents, and the second order, those inner neurons that are going to take it up to the brain, to the thalamus, we get a block. That's what presynaptic inhibition of the second order neuron means. During this as well, we talked about that central control theory and the central control trigger theory. The A betas and the A deltas and Cs are also stimulating inner neurons within that little SG area, specifically one that we call the enkephalin inner neuron, which is going to help either inhibit or facilitate the T cell as well. So again, and that's kind of that little bit of a memory piece. Like if we're really overstimulating one of these fibers more often, we're going to make it easier or harder to kind of open or close the gate. Level 1 pain control is really good for acute pain. Okay, if we remember that quick acute pain tends to really travel on those A delta fibers. And so this blocks those A deltas fairly well as well as the Cs. It's not very good for chronic pain because it's something that has to be constantly stimulated. You only get treatment, you only get pain relief during the time that the A betas are active. And so again, if we go back to rubbing your arm, you only really feel the stimulation while you're rubbing the arm, and you'd have to sit there and rub your arm all the time if you had chronic pain. Okay, but acute pain, if you do that for a while, then our body can start to get used to it. Maybe some other parts of the inflammatory process take over and the pain can subside by the time you're done rubbing your arm. But it's really more so for short-term pain relief. Okay, you guys are going to make fun of me. This is my little diagram to help explain how the gait theory level 1 pain control works. Okay. So we have A beta, right, those large fast fibers. We have our A delta and C fibers, which are smaller and slower. So if we stimulate those A delta fibers, the pain signal comes through the T cells. So again, this is happening in the spinal cord at the SG. The signal is allowed to pass. There's no closed gate. It goes up our spinal thalamic tract up to the thalamus, and that would then go with third order neurons up to the sensory cortex, and we would experience pain. So ouch, right? Stimulate, we sense pain. Now what we can do, okay, they're keeping the gate open right now. Those small fibers are trying to keep the gate open, and right now they're the tract with the most activity. So they're prevailing, so that signal is going up. Now if we throw in stimulating our A beta fibers, let's see what happens. So we stimulate the A beta fibers, that stimulates the gate to close on the A delta and C fibers, and now we don't feel pain anymore. The ouch goes away. Now we're just feeling that sensation. So again, that could be the touch of rubbing the arm. When we start talking about E-stim, it's going to be the sensation that we create with the electricity. But basically that inhibition occurs based on the activity of the A beta cells. One video I've attached on the Moodle site some additional videos by other people. So if you need a different perspective on this, if what I'm saying isn't making sense, just so you know, one of those videos calls the T cell actually like the P interneuron for projection, the projection one that lets it go up to the brain, project up. So don't get confused with that. They're just calling it a P. We call it a T. They mean the same thing. Okay. So this was part of that story, right? This is the ascending mechanism, the A betas. But we also talked about that part with the enkephalin interneuron, right? And that we're constantly getting feedback, the central control trigger kind of aspect of things. So additional inhibition can occur when the large diameters synapses with the spinal enkephalin interneuron. So that signal can also activate an enkephalin interneuron, and then that would release enkephalin all within the spinal cord. And enkephalin inhibits those small fibers even more, so can close the gate even faster or more adamantly on those smaller fibers. So this is what this diagram is going to say. So the substantia gelatinosa is stimulated by those nerve activations, as well as the enkephalin interneuron. And then enkephalin gets released, and both of those can either help close the gate faster or slower, depending on what the body is used to, what the history of the situation is, and so on. So now we're going to throw the SG in there. Again, we stimulate our A delta fibers, we sense pain. If we stimulate our A beta fibers, which also stimulates the SG, and we're stimulating those more so than the A deltas, so our body has more feedback from the A betas, then the gate closes even faster than it did on the last diagram, and the ouch goes away. It's more efficient. Same thing with the enkephalin interneuron. So again, we stimulate, we feel pain, enkephalin gets released, that closes the gate faster again, and we get more effect. Okay, so all of that information keeps coming into those two gateway and kind of feedback mechanisms, and they can facilitate or inhibit the gate. In this case, we're showing it facilitating the gate, closing the gate faster on those A deltas. So just to kind of see it again, this would be an inhibitory one, right? So if we're not used to that, it closed a little bit slower, it took a little bit longer, we had a little more experience of the pain. Okay, so that was our ascending mechanism. That was our level 1. Now we're going to go into our descending mechanisms. Okay, so all that was taking place in the periphery. The enkephalin was happening in the periphery, the substantia gelatinosa, all that feedback was happening from the periphery, just kind of reading which nerves get stimulated most often. The A betas, blocking the gate, all that was ascending. So now to our descending, which is going to help answer some of the holes in the gate theory, right? Like, why does the brain get involved? What are endorphins? How does that affect things? Okay, so the cognitive control gets answered here. And you're going to see we're no longer going to be talking about those A beta fibers. So our level 2, what we call central biasing. This is the concept of using pain to inhibit pain. And that sounds scarier than it is, especially when we start talking about using pain, creating E-stim signals that are slightly more painful to block the pain that the person's in. But we'll talk about its benefits. So the idea being, if we bombard those A delta and C fibers, specifically in this case the C fibers is our target, but the A deltas come along for the ride because they get targeted in the same manner and respond easier than the C fibers even do. So in this case, we're trying to bombard those fibers in order to trick the brain into not recognizing the actual pain, but to feel this sensation and to then have a response to that. Okay, So the central biasing theory of pain control uses that descending mechanism of pain relief. So within the cerebral cortex, specifically what we call the aqueductal gray region, that's going to activate what we call the Ralphie nuclei, and that's going to initiate this mechanism. And so again, descending mechanism, we stimulate so much pain going up to the brain that the brain needs to respond because the body's just in so much pain. So it's going to come up with a way to try to inhibit some of that pain. And that's what this takes place. So eventually the periaqueductal gray matter activates the Ralphie nucleus, or the RN, and that sends info to release serotonin. So serotonin is released at the spinal level. So we're still not in the bloodstream like we will be in our next theory. This is all happening within the spinal cord itself and the periphery, the central nervous system. So serotonin is released at the spinal level, and that's going to inhibit the second order neurons. So it's going to essentially close the gate. It's also going to activate that spinal enkephalin interneuron. So the enkephalin is going to get released. That's going to help close the gate. So the serotonin actually stimulates the enkephalin interneuron. And enkephalin and norepinephrine, again, all just within the nervous system. We're not talking blood flow yet, within the nervous system. And that's going to inhibit further T cell activation from there. So the brain is actually facilitating the closing of the gate and the inhibition of those small pain fibers. Benefits, right? There has to be a benefit, or why would you want to use more pain to inhibit pain? You get longer lasting results. So instead of like the gait control, the level 1, where you only got results as long as that stimulation was present, as long as we kept rubbing our arm, or as long as we were using the e-stim. In this case, because we release the serotonin and the norepinephrine within the spinal cord, it will keep the gate closed longer, potentially 1 to 3 hours after the treatment. Okay, it won't feel great during the treatment, but it usually feels better longer after the treatment. It's by far the most painful stimulation, so it's really not good for acute injuries because the person's already in so much pain that, you know, we don't want to initiate so much pain that it's kind of unbearable. We want the person to trust us. Also, if they're in that acute pain, they should be resting and doing a lot of other things too. So we don't necessarily need this 1 to 3 hour pain relief, because everything we're doing during that early stages of the inflammatory process is about pain relief and swelling control and all those things we talked about in chapter 1. Okay, diagram. It's going to look a little bit different as we have now left behind those A beta fibers. But we still have the enkephalin interneuron, we still have the T cell, and we have the A delta and C fibers, our pain primary afferents, right? So same thing, we stimulate those from a nociceptor, we sense pain, okay? What central biasing is saying is, let's stimulate that more and more. So think about more and more of these same lightning bolts coming at the A delta and C fibers. Specifically, again, we're really trying to target those C fibers in this case, but the A deltas are just kind of along for the ride. So we're targeting as much of those C fibers as we can. What happens is we get serotonin to release. That tells the enkephalin and norepinephrine to be released to affect the enkephalin interneuron, which is then going to close the gate. So A betas are not involved, okay? It's a different mechanism to get the gate closed, okay? So let's watch it one more time. So we stimulate the A deltas and C fibers, we experience pain. If we keep stimulating more and more and more and more, eventually the Ralphie nuclei will tell serotonin to release, which will tell enkephalin to release, which will affect the enkephalin interneuron and close the gate. So level 2 pain control right there. Our level 3 and our final is also named androgynous opiate release. And so this is really that descending mechanism of the gait theory that we talked about, of how do we explain the use of endorphins and things like that. So in this case, when we talk about creating that with e-stim, we talk about using strong rhythmic muscle firing to stimulate the A delta fibers. There's other ways that androgynous opiates get released. Our parasympathetic and sympathetic nervous system can release them, but this is one way we can do that without kind of having to go through that mechanism. Strong rhythmic muscle firing ends up stimulating our A delta fibers specifically. So level 1 was A betas, level 2 was really C fibers, A deltas kind of along for the ride, and this one really targets our A delta fibers. So in this case, the A delta fibers activate our T cell, okay? So again, we're talking about it being a descending mechanism, so the brain and the higher centers of the brain are going to be involved, okay? Specifically in this case, the hypothalamus, because it controls pain, especially pain that's caused by prolonged intense stimulation. So again, if we do a length of time with this really intense stimulation of those A delta fibers, that's going to stimulate the hypothalamus to get involved. And then the hypothalamus is going to tell the anterior pituitary gland to release the beta endorphins. That's going to then stimulate the periaqueductal gray matter, which is going to stimulate the Relfé nuclei, which is then going to stimulate the enkephalin and or the serotonin release, which is going to affect the enkephalin and norepinephrine release. And it's going to follow that same kind of pathway. The difference being, main difference being here, is that these beta endorphins don't just stay in the spinal cord like the serotonin and the enkephalin did. They're transmitting throughout the whole body. And they're going to, they're neurotransmitters, so they're going to sit at different receptors on different cells and have different other responses in your body. So norepinephrine transmitting throughout your body is going to also affect your heart rate and affect other things. Now we keep it on a level low enough that we don't see those results, but it's going to have these longer lasting effects because it's out in your bloodstream. And until they get picked up, until your body can flush it completely out of your bloodstream, they're constantly around. Okay, so again, we get that long-lasting 1 to 3 hours. And we also usually have general sensations of well-being. Norepinephrine kind of does that as well, because again, these chemicals are going to affect other parts of your body, not just within the spinal column and the central nervous system. It's definitely less painful than level 2. It's still not the best for acute pain, because in order to get it, we have to create that muscle contraction. And if someone's in the inflammatory response phase, that goes against our police, right? The protection and kind of the controlled movement idea, or the stabilization. You know, we don't want to create this kind of unnecessary muscle fiber contractions that could then pull on areas that are just healing, that don't have good solid scars and type 1 collagen fiber and all of that stuff. So we tend to use it more for chronic or conditions that are out of the inflammatory response phase, so maybe have moved into the proliferation or repair phase a little bit more. But it's definitely less painful than level 2, but it takes longer. It takes a longer treatment because, like I said, it really doesn't get stimulated. The hypothalamus doesn't get stimulated until kind of a prolonged, intense stimulation. Okay. So let's look at our diagram. It looks a little more complex because there's a few more steps, but it's essentially the same. So again, stimulating, we feel pain. Now the pituitary releases the beta endorphins. That then goes and affects the PAG and the RN, which releases serotonin, which is then going to release the enkephalin to the interneuron and eventually close the gate. But realizing that these beta endorphins that got to the periaqueduct matter and the Relfé nuclei are also floating around in the body. And so as long as they're out in the bloodstream, this gate stays closed. So that's how we get that 1 to 3 hours. So let's look again. We stimulate the A, delta, and C fibers, and we sense pain. Okay? Now, if we keep stimulating the A delta fibers specifically, that's how we'll get this response. So we go to the reticular formation, which sends the signal to the pituitary gland, which releases the beta endorphins, that sends out that signal to release serotonin, which is going to release the enkephalin, which is going to affect the enkephalin interneuron and close the gate. So, review these. Okay? These are going to come back when we start talking about eStim. This is by far the most complicated part of this chapter. So watch this video a couple times, go and look at the other videos that I've posted, read in your book, find your own videos, whatever you need to do to understand this. Make sure you grasp this concept, because this is the basis of us moving forward and starting to talk about eSTEM, which is going to be our first modality that we're going to talk about in a couple weeks.